Welcome, NEET aspirants. This question is from the chapter Locomotion and Movement, and it is on muscle contraction. So let's attempt the question first. Select the correct statements regarding mechanism of muscle contraction. It is initiated by a signal sent by CNS, correct, via sensory neuron. Sensory neuron is incorrect, so strike out A. A is incorrect. Then neurotransmitter generates action potential in the sarcolemma. Correct. B is correct. So these two will be the answer. Rule this thing out. Now we have to, E is common in both option 1 and 4. So we have to rule out C and D. Increased calcium level leads to the binding of calcium with troponin on actin filaments. This is correct. So let's rule out D. Masking of active site for actin is activated. This is a wrong statement. So confirmed answer is option 1. Mark it on OMR sheet. 197 is 1. And we are done with this question in 1 minute 5 seconds. So let's understand each and every statement asked in this question. First one. It is initiated by a signal sent by CNS via sensory neuron. So sensory neuron carries the signal towards CNS, and signal from CNS is transmitted by motor neuron. And this is the reason this statement becomes incorrect. So CNS, it sends the signal for muscle contraction via motor neuron, and this activates the muscle fiber. So motor neuron and muscle fiber together constitute a motor unit. Then, second statement says neurotransmitter generates action potential in the sarcolemma. Sarcolemma is the membrane of muscle cell or muscle fiber. So, this statement is correct. So, see, the junction between the motor neuron and the muscle fiber is called neuromuscular junction. It is also called motor end plate. So, the neurotransmitter is acetylcholine and it generates action potential. So, this is acetylcholine, the neurotransmitter molecule. When it binds the receptors on sarcolemma, it is going to depolarize. That means it will generate action potential on sarcolemma. So, this statement is correct. Next statement says increased calcium level leads to the binding of calcium with troponin on actin filaments. This is correct. If you want to visualize it, so these globular structures, these are actin filaments, and then you can see tropomyosin, and these complex proteins are troponin. Sarcoplasmic reticulum releases calcium, and these calcium ions bind to troponin, and this helps in removing the masking of active sites for myosin. The next statement says masking of active site for actin is activated. This is completely incorrect statement. I'll show you why this is incorrect. So here you see a monomer of myosin. It is called meromyosin. So this region, globular region, is called head, and this is a short arm. Together head and arm are called heavy meromyosin. And this region is called tail or LMM, which stands for light meromyosin. So what you see here is actin binding site. What is this? This is ATP binding site. It has ATPase activity. So there is nothing which covers the actin binding site. Okay. And this is the reason the statement masking of active site for actin is activated. This is completely wrong. On the other hand, the right statement would be unmasking of active site, and instead of actin, it should be myosin is activated. So unmasking of active site for myosin is activated. This is the correct statement. So as you saw in the earlier image, calcium binds to troponin and it displaces, it causes conformational change in tropomyosin, and thereby it exposes the myosin binding sites on actin. The last statement says utilizing the energy from ATP hydrolysis to form cross bridge. Correct. So see, the myosin head has ATP binding site. So due to ATP hydrolysis, myosin heads come into a cocked position, and then only the interaction with actin is possible, which in turn leads to cross bridge formation. So utilizing the energy from ATP hydrolysis to form cross bridge, this is a correct statement. So let's understand the structure of muscle to understand how muscle contraction occurs. So first I will explain you what are actin filaments. Actin filaments are called the thin filaments of muscle. So this globular actin is called G-actin. Then it polymerizes this way to form a filament. This is called F-actin. Okay, filamentous actin. And then you will have another filament. They are wrapped around to form a double helix. Okay, and this becomes the actin thin filament. So actin filaments are also associated with tropomyosin. So you can see these two strands that are wrapped around actin. These are tropomyosin. And a complex protein called troponin, the black beads over here, these are distributed at regular intervals on tropomyosin. So this is going to mask the binding site for myosin on actin filaments. So this is how muscle contraction is controlled. The masking will block myosin heads from engaging with the actin filaments. So what happens when action potential depolarizes sarcolemma? This causes release of calcium ions from the sarcoplasmic reticulum. This is a type of smooth endoplasmic reticulum. So upon activation by action potential, it will start releasing calcium ions. Okay. And these calcium ions will bind to troponin. So you can see here, binding of calcium to troponin has caused conformational change in the tropomyosin, and it exposes the myosin binding site on above and below the actin filaments. So myosin are arranged this way. It has two heads, so it can easily interact with both sides of actin filament. Now, in order to understand how myosin heads pull actin filaments, watch this structure. First things first, this is how the myosin are arranged. Okay, in the earlier diagram, I showed you a single myosin monomer unit. In the sarcomere, many myosin proteins are arranged this way. Okay, so the heads are spaced out. So the region where the myosin filaments are present is called A band. In the center, there is this M line which holds the myosin filaments in place. H zone means there will not be any overlapping actin filaments in this region. But at the time of contraction, the actin filaments will extend deep here and the H zone almost disappears. So in the NCERT textbook, you will not find this term M line. Instead, center of A band is mentioned there. So here A stands for anisotropic or the dark band. I band is called the light band. I stands for isotropic, and this consists of the light actin filaments. So in the center you can see Z line. This Z line holds actin filaments in place. And this is the center of I band. So now let's see how myosin and actin are arranged in a sarcomere. So sarcomere is the functional unit of muscle and it extends from one Z line to the next Z line. So Z line has these actin filaments extended out. Okay. And in the center there is A band which has myosin filaments. This is the region where there is overlap of myosin and actin. Okay. This region which is marked as H zone, there are no overlap of actin. This has only myosin filaments. So this is the resting position. You can see clearly H zone where there is no overlap of actin over here also. And if you notice the distance between Z line, they are far apart. And upon contraction, see the Z line comes closer and the H zone completely disappears. So sliding of thin filament over thick filament helps in the contraction. And during contraction, the Z line, they come closer. So shortening of the sarcomere occurs. So here you can clearly see the actin filaments, the thin filaments have completely overlapped the thick filaments. So they slide in towards the center of A band. And once the calcium again moves back into the sarcoplasmic reticulum, the troponin hides all the myosin binding sites on actin. So these filaments, thin filaments, they slide back into the original position, and that is called relaxation of the muscle. So according to sliding filament theory, the first step is ATP hydrolysis. This changes the angle of myosin head into cocked position, the one which we saw here. And then only it comes close to actin and it can form cross bridges. So second step is cross bridge formation. Myosin head via actin binding site binds to actin filament. And then there is sliding or rotation of the myosin head. And this sliding pulls the actin filament towards the center of A band. And this causes shortening of sarcomere, and this is called contraction. So muscle contraction is associated with so many events. And then after this, a new ATP binds and the cross bridge is broken. So these four steps are repeated till calcium ions are pumped back to sarcoplasmic reticulum. Actins slide back to the original position, and this is called muscle relaxation. So these are all the steps of sliding filament theory. So this is all about this question. I hope this video was helpful to you. Thank you so much for watching my video.